I think it's had a substantial impact across the entire process, right from the real early stage discovery, what is the mechanism, if you look at things like spatial and the whole transcriptomics world, there's very fundamental questions about what's going on and developing therapeutic pathways. Then you start to look out maybe further down the process where you're actually talking about library screening. You've got a putative target and now you say, well, can we find that within a certain population? Immediately the use of single cell there. But what's quite interesting beyond that, of course, is you've got the whole next generation of therapeutics coming through. So beyond biologics, when you start to talk cell therapies, where the cell itself is the therapy, so then you've kind of almost extended the process. And of course, by its very nature, single cell technologies become very applicable into cell therapy, even into things like CMC and manufacturing and quality control, which takes it literally across the entire process from that very first what is my mechanism right through to have I made the right therapy and can I put it into a patient. So I find that very interesting, that breadth of application of single cell now, which I just don't think we had a few years ago. The first one, I'm going to channel the spirit of Richard Feynman here. There's size, his famous lecture about there's plenty of room at the bottom. The challenge I still see in single cell is the cells are at micron scales, but we think in terms of automation at millimetre or even metre scales. I keep thinking we ought to put a metric of goodness on the automation, on the ratio of size of thing we're measuring to thing we're using to measure. And there's orders of magnitude differences there, which kind of get a bit crazy. So the big thing for me about accessibility is ultra miniaturization. Let's get down to the scale of the cell, and that's the work we do. We do droplet work. That's really trying to push towards that, of actually working at the scale you need to address the scale and the speed challenges that then come. So that's a big one for me about getting the accessibility, is the size of it, and then making it more just accessible for biologists, not engineers. It's very easy to build automation that if you're an engineer you think it's great, and the poor old biologist just doesn't know even where to start. And that's no criticism of biologists. You don't need to know that stuff. You shouldn't know that stuff. So there's still a big need for just the almost more basic reliability, ease of use. Just that, don't worry about what's going on, just trust it and use it. If you bring those two together, ultra miniaturization and working at the biological scale, and that ease of use and it's kind of eliminating the engineering problems, those are the two real big ones for me. It is going to be pretty fundamental, but the way I think about it is the need for data in the AI world. So you've clearly got these new tools appearing, this idea that you can build, train a set of software, an AI model to predict stuff, undoubtedly true. The challenge then becomes, well, you've got to feed it. It's only as good as the data you can put into it to allow it to, do what it needs to do. And so you can immediately see that correlation with the single cell world, because how are you going to provide all this data? And not only that, how do you provide it at a workable dollars per data point? At some point you've got to get the data so cheap to gather that you can truly feed these models. So I think that's the fundamental interaction between single cell and AI, is that question of very high quality, very low cost data, and building the systems, the mechanisms to truly close the loop. The two feed off of each other in a very symbiotic way, and it is going to be utterly fundamental, because we are going to go that way, to using these kind of models. That's no different from what's happened in other industries, over many years. I mean, going back to the Manhattan projects, when they were doing the original, individual work there, they started using computers to try and predict what was going to happen. And other industries have done it for many years. And I used to work in Formula One. We use computers widely to predict stuff. You just do it because they can. But that's not magic. That's something that's been learned and introduced over many years. And I'm seeing a similar process appearing here, that you're just integrating these new models into your ways of working. They're AI rather more predictive models, but that's not really the point. The point is they're coming, they're very real, they will become part of it. And there's this very strong relationship between the world of single cell and data and these models to actually make a workable pair, an outcome that truly adds value. Ultimately, I get a new therapy at the end of it. We talked about usability earlier, and I still think that's a big part of it. You know, people don't want to have to learn an entirely new set of skills and disciplines just to run this device. I think stuff is still a bit big and ugly and awkward. You know, we've got this big dedicated lab full of very expensive machinery. Again, maybe going back to that theme of miniaturization and kind of removing that scariness from it. The data piece is fascinating. Again, back to what you're saying about AI, fundamental symbiotic relationship there. There is a real challenge around data and getting it in standardized, easy to manipulate forms, bringing up people's understanding of data analysis tools and how those kind of work. Maybe there's an educational part there that people have to just know how to do some of this stuff in maybe a way they don't now. But one of the other, talking about data, I think one of the other challenges to adoption, this is an interesting one, will become the value of the data in of itself. So I've just been to a couple of talks here at SLAS already, and already I'm hearing we get much better results with our AI using our data. Okay, so that would start to suggest the data itself has a real value to it. So how do you then correlate that with the need to share data, the greater good of having that consistent and standardized data? I think there's some very, very interesting conversations and thinking to be done there about how you manage both the sort of proprietary data and the commercial imperative, and yet the need to share data in order to build these models and truly move forward. So there's some other challenges to adoption which are maybe less about the science and the engineering, but more about society and economics and how you bring these things together. Yeah, no, there's some really exciting stuff happening. I mean, the whole world of spatial has been just brilliant to see all that coming through, and these ideas about how you can truly start to map the morphology and the spatial interactions. I think the thing I'm seeing coming, and it's kind of a maybe slightly odd answer, is that it's a lot more than single cells. So we talk about single cell, but ultimately biology does not exist as single cells. You know, I am not single, I am a set of cells. And so I think where we're going is that recognition of interaction, cell-cell interaction, the functions between cells. Now, a lot of the tools we talk about for single cell, you can start to apply to pairs of cells, or maybe you start talking things like organoids, you know, small clusters of cells. So I think, in my mind, that is one of the very interesting directions in the future about where this idea of single cell technologies can start to be applied into that more functional interaction between cells, whilst maintaining the miniaturization, the ease of use and so forth, but really starting to extend beyond true single cell, which is a little bit of an artificial system. So that's the one for me. How do we go beyond single cell to true functional interactions between cells and characterize those?